Glue-like proteins called cohesins are used by cells to keep DNA molecules stable and to repair damaged strands. Cohesins also play several important roles in chromosomes during cell division. Now, lead author Jill Heidinger-Pauly, along with colleagues at Carnegie's Department of Embryology, has found for the first time that cohesins are needed in different concentrations for their different functions. The research was made possible by a new technique which enables scientists to gradually reduce the concentration of a protein in living cells. A cell has a four-phase life cycle: growth, synthesis, growth, and mitosis. During the synthesis phase, DNA inside the cell's nucleus duplicates, resulting in two identical daughter chromosomes called sister chromatids. These twins must remain connected until the cell is ready to divide. This moment occurs in the last step of the cell cycle, the mitosis phase. During mitosis, cohesins play a role as chromosomes condense. Then fibrous structures called spindles form and attach to the chromosomes. Cohesin keeps the sisters properly glued until it is time for the spindles to pull the sisters to opposite sides of the cell. The cell then separates into genetically identical twins. The research revealed that DNA stability and repair, as well as chromosome condensation, were compromised by decreasing cohesion to 30% of normal levels. However, sister chromatid cohesion and chromosome segregation were not affected, even with levels at only 13% of normal. Normally, cohesin binds to regions throughout chromosomes, but the scientists found that when cells only had a small amount of cohesin, it preferentially binds to the center of chromosomes. They didn't know that this hierarchy existed before, and it helps explain why some cohesin functions might be more affected than others. The research is published in the online May 6th and print May 25th, 2010 editions of Current Biology. Dominic Pampano of Carnegie's Geophysical Laboratory has proposed a model that links phosphorus levels in the oceans with the first evolution of complex life forms. Rising levels of phosphorus would have caused algal blooms that pumped extra oxygen into the atmosphere, allowing complex organisms to evolve. The research focuses on the Proterozoic era, which extended from 2.5 billion to 540 million years ago. At the beginning of this era, the Earth's atmosphere may have been oxygen-poor and methane-rich, giving the sky an orange tint. Oxygen levels are thought to have increased dramatically during the Proterozoic. During the Great Oxidation event, ranging from 2.5 to 2 billion years ago, oxygen levels rose to 10% of their current levels. Continental rifting and glaciation during the same time could have changed erosion patterns, increasing the amount of phosphorus washed into the oceans. Pampano found that abundant phosphate deposits were laid down at the same time as the atmospheric changes. Single-celled organisms grew larger during this time and acquired cell structures called mitochondria. These so-called powerhouses of cells burn oxygen to yield energy. The second phase of oxygen rise occurred between about 1 billion and 540 million years ago and brought oxygen levels to near present levels. This period also experienced continental rifting, glaciation, and increased levels of phosphorus in the oceans. The earliest fossils of multicelled organisms appeared at this time, which climaxed with a spectacular increase of fossil diversity known as the Cambrian explosion. Over geological timescales, the effect on marine life would have been similar to that of high phosphorus fertilizers washed into bodies of water today, such as the Chesapeake Bay. There, massive algal blooms have had a widespread impact. The creatures of the Proterozoic had millions of years to adapt, but the quick changes being caused today may lead to significant extinctions. The study is published in the journal Astrobiology. This is John Strom for the Carnegie Institution.